Hello everyone. Today we'll discuss bioethanol production using yeast fermentation, a common method for producing ethanol fuel. In this process, the yeast Saccharomyces cerevisiae aerobically converts sugars into carbon dioxide with ethanol as a byproduct. Specifically, our team investigated how varying sugar concentrations impacts the rate of ethanol production, with constants small and large K being our ultimate goals. Through fermentation, glucose is converted into ethanol, lactic acid, or hydrogen. The final reaction for ethanol production from glucose occurs when each glucose molecule is converted into 2 molecules of ethanol and 2 molecules of carbon dioxide. This reaction takes place under anaerobic conditions and is catalyzed by enzymes in yeast cells. The efficiency and rate of this fermentation depend heavily on factors like temperature, yeast concentration, and sugar concentration. By studying these factors, we can better understand how to optimize ethanol production for industrial applications.
Our objective was to determine how varying the initial sugar concentration affects the rate of ethanol production, while keeping yeast concentration and the temperature constant. We used different sugar concentrations ranging from 15 to 25 grams and kept the yeast at 9 grams across all trials, with a constant temperature of 30 degrees Celsius maintained using a water bath. We used the following materials and equipment for this experiment: 4 glass bottles of similar size, 4 balloons to capture the carbon dioxide gas produced, a digital scale to accurately measure yeast and sugar, a paper ruler to measure the circumference of the balloons, and a water bath held constant at 30 degrees Celsius.
Our procedure involved the following steps. We weighed out 15, 18.3, 21.6, and 25 grams of sugar for each bottle, creating different concentrations. We then measured 9 grams of yeast for each bottle, maintaining consistency across all trials to isolate sugar concentration as the variable. Next, we added 150 ml of water to each bottle, creating a solution that facilitated fermentation. To keep the system anaerobic, we placed balloons on the bottle's mouth to capture the CO2 produced. We placed all bottles in a water bath held at 30 degrees. Every 3 minutes, we measured each balloon's circumference with a paper ruler to gauge CO2 production over time. We repeated each experiment 3 times for each sugar concentration to ensure accuracy in our data. By comparing balloon circumferences over time, we could assess how the different sugar concentrations affected the fermentation rate.
With our data collected, we aimed to calculate the kinetic constants small k and big K, which describe the reaction rate's behavior as a function of substrate concentration. The rate of ethanol production in our system can be modeled using a Michaelis-Menten-like expression adapted for fermentation, where R is the rate of ethanol production, C sugar is the concentration of sugar, small k is the rate constant, and large K is the saturation constant, indicating when the reaction rate approaches a maximum. By plotting the rate of CO2 production against the initial sugar concentration, we could determine the constant small k and big K through linearized methods like the Lineweaver-Burk or Eddie-Hofstee plots. These approaches helped us analyze how effectively yeast ferments sugars and where substrate saturation occurs.
In summary, our experiment explored how varying sugar concentrations influence bioethanol production through yeast fermentation. We observed differences in fermentation rates, indicated by the balloon inflation, which corresponds to CO2 production. By examining CO2 generation and applying kinetic modeling, we aimed to determine the rate constants K and K, which will be useful for scaling bioethanol production. Thank you for your attention.